Hi Year 12, just going to make a quick video on the light dependent reaction, which will hopefully help you understand what's going on. So first of all, you need to know the location of the light dependent reaction. It takes place in the thylakoid membranes of the chloroplast, so not in the lumen itself, in the actual thylakoid membranes, and you should know what the thylakoid membrane is. So there's two other parts that are embedded within the thylakoid membrane that you need to be familiar with, and those are called photosystems. So you've got photosystem 1 and then photosystem 2. Photosystem 1 was obviously discovered first, followed by photosystem 2. And at the centre of these photosystems, you will find a chlorophyll. And that's really important. We'll see that in a second. So if we were to zoom into a leaf, you can see you've got a plant cell. And then if we zoom into the plant cell, into a chloroplast in particular, you will see your thylakoids all stacked up on top of each other, making a grana. And then if we zoom into one of those thylakoid membranes, you can see, hopefully, what a photosystem looks like. And then you've obviously got your chlorophyll in the centre, which is in blue on this slide. So what happens is the chlorophyll will absorb light energy, which you can see happening on the picture on the right. So first of all, we're going to have a look at the following three processes that take place in the light dependent reaction. You've got non-cyclic photophosphorylation, cyclic photophosphorylation, and photolysis. Now you should already know what photophosphorylation is. It is using light energy to combine an inorganic phosphate to a molecule of ADP. So therefore, you obviously end up with a molecule of ATP. So let's first of all have a look at non-cyclic photophosphorylation. This is a nice little summary here. Now, obviously, like I said, photosystem 1 was discovered first, and then photosystem 2 was discovered after that, which is a little bit annoying because it would have been nice if they discovered photosystem 1 first, because that's obviously where we're going to start our little story here. So, with non-cyclic photophosphorylation, the first thing that happens is you have light energy that is being absorbed by your chloroplast, by your chlorophyll, sorry. And when that light energy is absorbed by that chlorophyll, it's going to excite a pair of electrons. And that pair of electrons will then be moved. Why is this not working? Okay, let's just go back a little bit. Okay, so that's weird. Let's have a look. The animation seems to be working. Here we go. It's working now. So your light energy, which you saw there, is going to be absorbed by the chlorophyll in photosystem 2, and that's going to cause a pair of electrons to leave the chlorophyll, and they're going to be excited to what we call a higher energy level. What happens then is that pair of electrons will be accepted by a molecule that's obviously found inside the membrane of the thylakoid. And then from that molecule, it's going to be passed on to another molecule, and then another one, and then another one, and then another one. So you're going to have a series of reduction and then oxidation along all these molecules and embedded within the thylakoid membrane. All of those molecules there make up what we call the electron transport chain. So you can see here the pair of electrons are being sent from one molecule to another, and then will be picked up by the chlorophyll in photosystem 1, which you can see where the cursor is just there. So those pair of electrons will be picked up. Now, as you can see, the pair of electrons, as they're being moved from one molecule to another, they're actually moving down the electron transport chain, and as they move down, energy is being released each time they get passed on, and it's that energy that's been released is going to be used to combine a molecule of ADP with an inorganic phosphate. So the energy is going to cause these two to join together to give you a molecule of ATP. So that energy is going to be stored in this final bond that joins those two together. So just to recap, light hits this part here, your chlorophyll. That excites a pair of electrons to a higher energy level, and then they get passed from molecule to molecule along the electron transport chain, and as they get passed they move down an energy level, and then that energy is going to be used to cause a molecule of ATP to combine with inorganic phosphate. And when you get to photosystem 1, the exact same thing is going to happen. Light energy is going to be absorbed by chlorophyll, and that's going to cause the pair of electrons to be excited to a higher energy level, and those obviously electrons that are coming down here are going to replenish the electrons that have been lost from the chlorophyll. So that allows this whole process to continue, a bit like a conveyor belt. Now these electrons here are then going to be passed on to a coenzyme, and that coenzyme is called NADP. So that molecule is going to pick up this pair of electrons. It's also going to pick up a pair of protons. We're going to have a look in a second where those protons are going to be coming from. So the NADP has picked up 2 electrons and it's picked up 2 protons, and you should already know that if you combine an electron and a protein you have hydrogen. So NADP is going to pick up a hydrogen, so it's going to be NADPH2, we obviously discussed that earlier, or I like to call it reduced NADP. And that reduced NADP will obviously be used in the next stage, which is our light independent reaction. But that's the first part. Now, I was talking obviously about the coenzyme NADP being able to pick up protons as well as electrons. But where are those protons coming from? So if we go back here, it's all to do with photolysis, the splitting of water. So we are now in the thylakoid lumen. So that's the middle part of our thylakoid. And this is what is happening. Light energy is actually going to split water, and you can see there that one molecule of water will be split into 2 protons, so obviously there'll be a proton there and a proton there, 2 electrons, so there'll be an electron there and an electron there, and it'll be split into your oxygen. Now the reason we have it as half O2 is because obviously there's only one oxygen there. Now oxygen doesn't exist as just a single atom because it's so volatile. So what will happen is it will quickly combine with another oxygen, because you've got to remember that there's loads of water molecules in this thylakoid lumen, so it'll be happening not just to one but to several. So those oxygen atoms will combine with another to make them a lot more stable, and that's why we write it as half O2, because we're trying to show that there's only one oxygen here. So half O2 is 1, which is why we write it like that. It sounds a bit strange, but those people who do chemistry will understand that. So what will happen then is that oxygen which has been created, it will move out of the chloroplast and then out of the cell and out into the environment. But it's these two that we're interested in. It's the proton and the electrons. So your electrons are going to obviously be transferred to photosystem 2, which is in the membrane of the thylakoid, so very close by, and then that's going to allow the electron transport chain to continue. And then it's these 2 protons, which we were talking about before, are going to be picked up by the coenzyme NADP, and when it picks up the protons, it will also pick up the electrons that have obviously flowed through the electron transport chain, and that is how it can actually pick up a hydrogen and then take it to the next stage. And then the final thing that I want to talk to you about then is cyclic photophosphorylation. So cyclic is obviously a cycle. Now this happens in photosystem 1. So obviously here we had photosystem 2, and then the pair of electrons get excited to a higher energy level and they move down the electron transport chain just here. But what can happen with photosystem 1 is the pair of electrons can obviously get excited, as we saw previously, and they move to a higher energy level. Now at this point they could be taken up by a coenzyme NADP to become reduced NADP, or they can actually flow back down the electron transport chain and regenerate the chlorophyll that they were originally left through excitation. So we can see that happening down there. And then obviously as it moves down the electron transport chain, energy will be released as it moves from one molecule to another, and it's that energy can be used to combine a molecule of ADP with an inorganic phosphate to make the ATP, which is important for the next stage, which we will be talking about in the next lesson. Then you put it all together and you've got a nice little summary that looks like that. So if you want to pause the video now and have a little look at that, and hopefully that will make some sort of sense to you. Okay, perfect.